learning objectives. At the end of this section, you all must be able to define what is microbiology, what is medical microbiology, and what are the important differences between the prokaryotic cells and the eukaryotic cells. Pathology has been divided into the different sections, including histopathology, hematology, and microbiology. There's another category also, that is chemical pathology, but that we will do along with all these: histopathology, hematology, and microbiology. So these are the three main sections we are going to do in pathology. Microbiology. We can divide this word into the three different words. Micro means small, bios means life or a living thing, then the logos means study. So microbiology is actually the study of small living things. These are so small that we cannot see them with the naked eye. We cannot actually see them without the help of microscope. So these are actually the microorganisms which can be unicellular or multicellular or acellular. Most of them are unicellular, but they are also multicellular and acellular too. History of microbiology. Antony von Leeuwenhoek is very important person who was the first microbiologist, and he gave knowledge about the bacteria in 17th century when he observed bacteria by using a single lens microscope of his own design. Louis Pasteur is another person who developed actually a process which is nowadays known as pasteurization, and this process was to kill the microbes. The pasteurization is actually accomplished by heating liquids to 63 to 65 degrees centigrade for 30 minutes, or heating liquids to 73 to 75 degrees centigrade for 15 seconds. Robert Koch was a pioneer in medical microbiology, and he worked in cholera, anthrax, and tuberculosis. All are very dangerous and important diseases. He was awarded a Nobel Prize in the 1905 for Koch's postulate. Actually, he set out criteria to test. Alexander Fleming, in 1929, he discovered penicillin. So thereby we became able to protect ourselves from the bacterial infections. Branches of microbiology. There are different branches of microbiology, including medical microbiology, industrial microbiology, food microbiology, soil microbiology, and the plant microbiology. All of these are equally very much important, but we are concerned with the medical microbiology. So we will do in detail the medical microbiology in our course. Medical microbiology. It is the study of causative organisms of the infectious disease of the human being and their reactions to the such infections. So in this we have to study the infective organisms, its infecting agents which are responsible for causing infectious diseases of the human being, and also the reactions, what they are causing actually, and also the response of the human beings against these infecting agents. It includes mostly etiology, that is the cause. Etiology is the other name for the cause of the anything. So here we are concerned with the cause of the infection. Then the pathogenesis, that is the mechanism of the disease, how the disease is occurring in the body, how it is actually starting and increasing and progressing in the body. Then epidemiology, Here we will see the how many people and of the how many areas are getting involved, are getting infected, and then the lab diagnosis. Here we actually have to detect the organism from the infected person, and for this we have to do some investigation, some tests. Then the treatment and the control of the infection. Once an individual becomes infected, then we have to control infection in that, and we have to treat that person to get relief from that infection. Major groups of pathogenic microorganisms. They have been divided into the two main groups, prokaryotes and eukaryotes. There is another third category, which is actually including acellular microorganisms, that is the viruses, which are pathogenic and quite distinct from the other organisms included in the prokaryotes and the eukaryotes. Prokaryotes are including only bacteria, and which are actually unicellular, while the eukaryotes are divided into the two subgroups, Protista and Metazoa. Protista are including fungi and the protozoa, while these are mostly unicellular. Metazoa are including helminths, and they are multicellular microorganisms. Medical microbiology is studied under the following heading: Pathology, that is the study of the bacteria. Whole aspects of the bacteria are studied under this heading. Then the virology, it is the study of the viruses. Mycology, this is the study of the fungi, including both the yeast and the mold. While then the parasitology, which is the study of the parasites, including all the parasites, including the protozoa as well as the metazoa. Then immunology, this is the study of the immunity, immune system of an individual. Host immune system in the normal cases as well as when any individual is getting infected. Actually, here we also see the deficiencies and the abnormalities occurring in the immune system. Congenitality are when these are acquired after birth. Then here you can compare different medically important organisms from each other. In case of actually cell, so cell is absent in the viruses, but it is present in all the other microbes, bacteria, fungi, and the parasites, all parasites, including the protozoa as well as the helminths, which are the metazoa. Then their size, which is about 0.0 to 2.2 micrometer in case of the viruses, while in the case of bacteria, it is about 1 to 5 micrometers. It is about the size of the RBCs of the human beings, so which is about 7 micrometer in size. So bacteria can reach up to the 7 micrometer too, while the fungi are having size about 3 to 10 micrometer. Especially yeast are more larger than the molds. Then the parasites, they can be about 15 to 25 micrometer. Trophozoites are the especially more larger than the cysts. These are the two different forms of the parasites. Then the nucleic acid, which is actually RNA and the DNA, they are present, both of them are present in the all microbes except the acellular viruses, in which either DNA is present or RNA will be present. Both cannot be present simultaneously. Then the type of the nucleus. As the viruses are acellular, so there will be no nucleus, but in case of bacteria, nucleus is of the prokaryotic type. That there will be only nucleoid but no nuclear membrane surrounding the nucleoid. While in case of the fungi and parasites, the nucleus is a true nucleus, nucleoid surrounding by nuclear membrane. While the ribosomes are again absent in the viruses, but they are present in the actually bacteria and the fungi and also in the parasites. But it is different in the prokaryotes from that which is present in the prokaryotes. So eukaryotic ribosomes are 88, while in case of the bacteria it is 78. Now the mitochondria are the other intracellular organelles like the Golgi apparatus that are absent in the bacteria, which is a cellular. Though it is cellular microbe, but these intracellular organelles are absent in case of the prokaryotic cells, but will be present in case of the eukaryotic cells. All the eukaryotic cells will contain these intracellular organelles, and the viruses again will not have because it is an acellular microbe. While the nature of the outer surface is actually also different with the different microbes. The viruses are having protein capsid and the lipoprotein envelope. Lipoprotein envelope is not present in the all viruses, but it is present in the some viruses, but it is absent in the others. While in case of the bacteria, the outer surface is actually rigid wall containing peptidoglycan. While the eukaryotic cells are devoid of the peptidoglycan, but in the case of fungi, there is rigid wall which is actually because of the presence of the chitin in the their cell wall. While the protozoa and the helminths, which are parasites, they are having flexible membrane. There is no peptidoglycan or chitin in the case of the parasite. As far as motility is concerned, viruses will be obviously non-motile, no cell, no flagella will be there. Bacteria, actually some bacteria are having flagella, so there will be those bacteria will be motile, but those which are not having flagella, they will be non-motile. While fungi are always non-motile, they will not have any flagellum, while all parasites not, but most of them are motile. Method of replication is also different. In case of viruses, they are not actually divided into different cells, but when they enter in the host cell, they are first disassembled, then they actually use the host cell machinery for protein synthesis and for the energy generation, and thereby actually they replicate. So they need host cell machinery to replicate. When they enter in the host cell, they replicate in the way that they deassemble, then they form their proteins, and their energy is generated by the host cell. Thereby they actually multiply their nucleic acids as well as the proteins and form multiple progeny cells by assembling these protein and the nucleic acids. While in case of the bacteria, they are dividing by the binary fission, and the fungi are multiply and replicate by the budding or the mitosis. Actually, yeasts are divided by budding, while molds are divided by mitosis. And in the case of parasites, they are all actually replicate or the multiply by the mitosis. The organism actually reproduce itself by complex sexual life cycles also. So the mostly helminths are cells is dividing by mitosis, while the others are actually producing themselves by complex sexual life cycle. Here's something which is actually this chart is for differentiating the prokaryotic cell from the eukaryotic cell. I just told you some different things which are actually present in the prokaryotic and eukaryotic cell, but this is a chart about the whole these things. Again, only between the eukaryotic and the prokaryotic cell. The characteristic points are same as the nucleus is important because it is nuclide in the case of prokaryotic cell, while in the eukaryotic cell it is the true nucleus because there the nuclear membrane is present surrounding the nucleus. Actually, the nuclide. The nucleus is again present in case of the eukaryotic cell, but will be not present here in the prokaryotic cell. Chromosome number is important. The prokaryotic cell has only single chromosome, which is circular, while the eukaryotic cells having more large number of the actually chromosomes, and they are linear, not circular. Mitotic division, I have told you, is present in case of the eukaryotic cell, but is absent in case of the prokaryotic cell. There is a binary fission. DNA-associated histones are absent in case of prokaryotic cells. Introns are absent in the prokaryotic cells, but they are present in case of eukaryotic cells. Then intracellular cytoplasmic organelles like mitochondria, lysosomes, Golgi apparatus, endoplasmic reticulum, all these are absent in case of the prokaryotic cell, but all these are present in the eukaryotic cell. So cytoplasmic organelles are present only in the eukaryotic cell. Then I have told you about the size of the ribosomes also. So it is also given again here that the 70S ribosomes are present in the prokaryotic cell, while 80S are present in the eukaryotic cell. Cell wall, as far as cell wall is concerned, the bacterial cell wall is consisting of peptidoglycan, and muramic acid is present in their cell wall. They are composed of in a such a way that they are consisting of muramic acid, while both these are absent in case of the eukaryotic cell, but the sterols in the cell membrane of the eukaryotic cell are present, while are absent in the bacteria except the one mycoplasma, which is a wall-less bacteria. So sterols are present in only the mycoplasma. Here you can see the diagram of the eukaryotic and the prokaryotic cells. So you can see here the nucleus inside is present inside the cytoplasm, and it is having actually nuclear pore, nuclear envelope, that is a nuclear membrane, and the chromatin nucleus is present there, chromatin is there, nucleus is there, ribosomes are there, plasma membrane, mitochondria, and the Golgi apparatus, lysosomes. These all are present here, which are the cytoplasmic organelles, and also smooth endoplasmic reticulum and the rough endoplasmic reticulum are present over there. Flagella is present, secretory vacuoles, cytoplasm, and surrounding plasma membrane is there also. But in case of the prokaryotic cells, most of the things are absent like the cytoplasmic organelles, but there is a cell wall, important cell wall is there, consisting of peptidoglycan. In case of the some actually prokaryotic cells, the bacteria, it is thick, very much thicker, but in case some it is thin. There is also outer membrane which is in some one category of the bacteria, but are absent the others. But bacterial flagellum is also there, which is also present in some of them, but not in all of the bacteria. Cytoplasm is there, 70S ribosomes are there, and the capsule surrounding the cell wall is also there, but not in the all cases. In the some bacteria it is present and making those bacteria more virulent because it protects the bacteria from the immune system of the host. DNA you can see is in the center as the nucleate because there is no nuclear membrane surrounding it. So this is the chromosome in the center. Then the one thing you can see is a mesosome. It's invagination, which is actually dividing the cell into the two during the binary fission process. So before the binary fission, this Division is completing the DNA and all the other actually structures are equally divided into the both progeny cells. As you can see, there's a big difference between the size of the bacterial cell and the animal or the eukaryotic cell. The eukaryotic cell is about 10 microns larger than the bacterial cell. Here you can see the size of the bacteria. The smallest bacteria, Microplasma, actually is about the size of the largest virus, that is the pox virus. That is about 1 micron. Bacteria are the smallest organism capable of surviving outside a host. The longest bacteria rods approach the size of some yeast in human red blood cells, 7 micrometers. These are the different microorganisms, the different sizes. Differences between the viruses and the bacteria are there also. Viruses are the obligate intracellular parasites. They cannot live outside the cell. Bacteria can live outside, but some are there like the rickettsia and the chlamydia. They are the parasites and they are living intracellularly. There is no ribosome in the virus, I have told you. Ribosomes are present here. DNA, RNA, both. One of them is present. DNA and RNA both are present in the bacteria, but not in case of viruses. Only one of them will be present. And viruses are so smaller that they cannot even seen by the light microscope, but they need electron microscope to be examined or detected. While the bacteria can be seen by the light microscope, though they are all of them are not visible with the naked eye. They are too small to see with the naked eye. So these are the main differences. A natural phylogeny is there in the case of bacteria, but the tangled phylogeny in the case of the viruses, and there are only 10 to hundreds of the genes in case of viruses, but there are hundreds to thousands of the genes in case of the bacteria. Nomenclature or terminology is very important. Bacteria, fungi, protozoa, and helminths are named according to the binomial lineal system, which uses the genus and the species. They are named by giving both the genus as well as the species. For example, Escherichia coli. It is a bacteria, gram-negative bacteria, which is actually having Escherichia genus and coli is the species. Then the Candida albicans. Candida is actually genus albicans is the species. So in this same genus, the different species can be. But the viruses are not so named because they are having single name, like poliovirus or measles virus. Some viruses named with the two words, like herpes simplex virus, but those do not represent their genus or the species. This is the whole name, the herpes simplex virus. Scope and advantages of the microbiology. It helps in diagnosis of the diseases by isolating and identifying the causative organisms from pathological lesions, and by doing the antibody tests. For example, ELISA or some other tests also. While second is the prognosis of the disease. For example, in case of the typhoid fever, rising titer signifies the active disease and ineffective treatment, while falling titer means curing disease and effective treatment. Third is that it helps in the guidance in treatment. For example, by doing culture sensitivity, we can culture the organism in pure form and can suggest effective and specific drug for the treatment of that particular infection. And thereby we can easily treat by giving the specific treatment to that person. Fourth thing is that it helps in finding out the source of the infection. For example, in certain outbreaks of the infectious disease, we can find out the source of the infection, and we'll try to actually destroy that source. And thereby we are actually defending the human being. We are saving them from getting the infection. So you can see the rod shape, spherical shape, and spiral shape, and also you can see in the next other pictures same thing. Rod, bacilli, cocci are the spheres, and the spirea are the spiral shaped organisms. So the pleomorphism is present in the bacterial shape. There will be some more changes, but the basic structure are this: cocci, rods, bacilli rods, spirochetes are spiral shaped. And these again are arranged in a different way. So they are named according to the shape and their arrangement. Coccus is rounded, Diplococcus when the two cells are joined, where Staphylococci when these cells are arranged in the clusters, grape-like clusters, while they are given name Streptococci when they are in chain. They are named Sarcina when they are 8 in number, and trade when they are 4 in number in one place. Again, you can see the shape and the arrangement. So this will be done in the next lecture in detail. You can see again, actually different arrangement is according to their actually division in the different directions. Here you can see the bacteria along with the red cells. Green cells are actually bacteria. So this is all about the introduction of microbiology. Now in the upcoming classes we'll do in detail